Hey everyone, welcome back to genetics. So we're so far going in through chapter 2, which is mainly involved in how genes are passed on from one generation to another. Last time what we talked about was how does a single gene get passed on from one generation to another. So a single gene. But today what we're going to do is we're going to do multiple genes now. So all we did last time was just big R, little r. We crossed them together and we use that to determine what the probability of getting a certain genotype or phenotype in the F1 or F2 generation. But today we're combining another gene, and multiple genes for that matter, into this process to figure out how we can determine what a certain genotype or phenotype frequency is. So to begin with, as we talked about last time, we talked about something called law of segregation. And the law of segregation was that you would have two copies of a single gene that are carried by an individual, and in those gametes from the individual, those alleles are split equally, so that way you have half of your gametes that contain one allele and the other half contain the other allele. But the law of independent assortment is another law generated from Mendel, and all it talks about is that you will have alleles of two or more genes they will segregate independently of each other such that all possible combinations of alleles in the gametes exists at approximately equal frequencies. So this is basically what the law of independent assortment is. It's essentially where you have multiple genes such as big A, and big P. These are two different genes. You can tell them apart by the difference in the letter. So if they have a different letter, that means they're different genes. And essentially what's going to happen here is that one of your gametes from this individual will be big A and big B. The other three will also be big A and big B, big A and big B, and big A and big B. And this is essentially what this is. It tells you that when you're crossing two individuals together that have two different genes, what you can use this, you can essentially cross each gene one at a time, and then you can combine those phenotypes or genotypes with the other gene cross to figure out a certain genotypic proportion or a phenotypic proportion. So from like last time what we did, we had an individual that was big A, little a, and we had another individual that was also big A, little a. So what we concluded last time was that in this situation, you would have approximately equal frequencies of gametes. So you would have a half chance to get big A here and a half chance to get little a. Parent number 2 would also have the same frequencies as well. And what you will end up with in a complete dominant situation, which we're still going over, is that you would have 1/4 of your offspring are big A, 1/4 big A, little a, 1/4 big A, little a, and then 1/4 little a, little a. So basically our genotypic proportion from this monohypercross is a 1 to 2 to 1. Essentially where we get, because big A, little a here is the same one as this one over here, we're going to combine them together by addition. So 1/4 plus 1/4 is 1/2. And then the phenotypic ratio is essentially, this phenotype is the same as the big A, little a phenotype. So it's essentially 3 to 1. So this is essentially what we're going to be doing as well in a cross. So let's say in a cross we have a situation where we have big A, little a, big B, little b, crossed with big A, little a, big B, little b. So in this situation, I'll tell you the little bit, like the normal human being way of how you can explain this is essentially what you're going to do is you're going to separate each of these genes into their own crosses. So what we're going to do first is we're going to cross the A's together with themselves, and then we're going to cross the B's together with themselves, and then after we're done with that, then we can combine them together into a single unit by multiplication. So hopefully that makes sense. And basically what we're doing here, the way how you're actually supposed to explain it is that, like how I said over here from the independent assortment, law of independent assortment, that each of the genes will independently assort away from one another. So basically you getting a phenotypic proportion from a big A, little a cross with a big A, little a is completely independent of any other gene. So basically you would always get a 3 to 1 ratio from big A, little a from that cross. And same thing goes towards the B genes as well. Doesn't matter what you have from A or B or C, you're always going to get that same proportion of 3 to 4 big B whatever phenotype, and then you would have a 1/4 chance of little b, little b. So what we're going to do right now is we're going to do the exact same thing, and what we're going to look for here is the phenotypic proportion. So what is the phenotypic ratio? Let's do that. So what we're going to do here is, like I said, we're going to cross big A, little a with big A, little a first, and always do them independently beforehand. Okay? And we're going to move away from the punnet squares for now. So let's start off. To get the phenotypic proportions from a big A, little a crossed with a big A, little a, it's essentially you would have a 3/4 chance to get big A whatever. They're the same phenotype. And then 1/4 chance to get little a, little a. Right? And then you would have, for the big B, little b genotype, or from the big B, little b gene, if you cross them together, you would also have a 3/4 chance to get big B whatever, and then 1/4 chance to get little b, little b. Okay? So what you can do now is you can cross them together one by one. So what we're going to do first is I'm going to draw these lines, and these lines represent what you should multiply together. So what we're going to do is we're going to combine the big A phenotype, and we're going to combine it with the big B phenotype. So basically right here, to get the big A whatever and big B whatever ratio would be 3/4 multiplied by 3/4, which is 9/16. Okay. So next, what we're gonna do is we're gonna erase that to get the big A phenotype and the little b phenotype. You write it down. To get big A and little b phenotype, it's essentially 3/4 multiplied by 1/4. That is 3/16. Okay. So now we're going to continue the pattern as well. Little a crossed with big B whatever. So little a, big B whatever phenotype is 1/4 multiplied by 3/4, which should be equal to 3/16. And then the last one, which is little a, little b, it's essentially 1/4 multiplied by 1/4 is 1/16. And these ratios right here that I've just calculated, this is referred to as a dihypercross. And what I've basically did, so you're crossing two individuals that are heterozygous for both genes, and you should get a ratio that is a 9:3 to 3:1 ratio. Okay? There are two ways how you can essentially do what I just did over here. The way how I just did it right now was what I refer to as the matrix method. So this is a method used so you can determine the ratio or the phenotypic proportions from a cross. But there's also another way how to do this as well. It's called the branching method. But it employs a similar method too. So the branching method is kind of similar. You're going to do the exact same thing what you did before. You're going to segregate the genes independently away from one another and cross them together. So big A little a with big A little a, you would have a 3/4 chance to get big A whatever, and then 1/4 chance to get little a little a. And then in this situation, what you would do is, since we have two products in the next cross, we would make two branches from each of these nodes, something that will look like this. And same thing for the other node as well. You would make two branches because we have two products. And then this is referred to as a node. The initial product that we had for gene A. So this method, what you can do now is you can just follow the lines and you would multiply across to get big A whatever, big B whatever. You would have a 9/16 probability. To get big A whatever, little b, 3/16. And then so on and so forth. Little a, big B whatever, you would have a 3/16. And then little a, little b, you have 1/16 probability. So we can do some more practice problems, but one of the things that you should know is that the branching method, one of the pros between this one is that one of the pros for this one is that it's reliable. In other words, you're writing down every single combination that you can on your paper, but it takes a very long time to draw out the branches. The matrix method, on the other hand, as you can tell, I only had 4 different things written down over here, and from those four things that I wrote it down, I got the answer pretty quickly. So the pros, this is pretty quick. But the cons, it can get a little confusing if you're not careful. And what we're going to do is I'm going to use both of these techniques in our practice problems so you can get a little bit of a hang of it. So let's do some practice problems right now where you have this following genotype and you have this one as well. And what you want to do is you want to figure out what the probability of the next offspring that has a phenotype as one of the parents. Looking at the parents, what can you tell? The first thing is that both parents are phenotypically dominant for all genes. Okay, so because of this, what you can do is we're looking for an individual that is completely dominant at every gene. Okay, so let's start off our crosses, and because we're looking at the phenotype proportions, we're only going to be looking at the phenotypic ratios instead of the genotypic ratios. So let's get us started on with that. So you were crossing big A little a with big A, or big A with big A little a. The phenotype proportion to get big A is simply 100% to get big A, whatever. Okay. To get big B little b with big B little b, the probability of you getting the big B phenotype is 3/4. And then 1/4 chance to get little b. Big C with big C little c, the probability of you getting a big C phenotype is 100%. And then now we're crossing big D little d with big D little d. The probability of you getting big D phenotype is essentially 3/4 big D and then 1/4 little d. And then the last one, big F with big F little f, the probability of you getting a big F phenotype is all 100% big F. So, because these guys right here are all 100%, we can ignore them. We don't have to include them in the calculation. And the only ones that we have to include is the B and D genes. But what is something that you can also determine from the question, that you can figure out a quick way how to figure out the phenotype proportion of the parent, of being completely dominant at every gene? One thing you can realize is that you don't actually necessarily have to calculate every single possibility in this situation, because the question is not asking you to figure out every possibility from this cross. The question is telling you to figure out the phenotype proportion of an individual that is completely dominant at every gene. So basically, we're not looking for the probability of you getting, big B whatever and little d. We're looking for a specific combination that is big A whatever, big B whatever, big C whatever, big D whatever, and big F whatever. So what we can do with this is that we can ignore these recessive individuals and we can just calculate all across to figure out this answer. So 1 times 3/4, 3/4 times, and we're going to ignore the C, we're going to ignore the A and the F because they're all 100%. So 3/4 times 3/4, that will lead us to a 9/16 chance of getting an individual that is big A whatever, big B whatever, big C whatever, big D whatever, and big F whatever. So basically that's all you have to do in that situation. So whenever you have something like that, and we're going to go over this in part B as well. But let's say you have, but now we have a different situation where the problem is asking you for a genotype, but the problem is asking for a very specific phenotype, which is that. So how do we figure that out? So we're going to do it one by one again, but we're only looking for a specific combination of a genotype, so we can skip all the other branches that we had to write because the question is asking for a specific combination. If the question just asks what is a genotypic ratio, then we would have to do every single combination using the matrix method or the branching method, whichever you prefer. Okay? So with this one, to figure out, let me erase this. We're looking at big A, little a first. We're crossing big A with big A, little a. And I recommend students to remember all the single gene crosses. That will save you some time on an exam. And I'll just tell you them right now. So to get big A crossed with big A, little a, the proportion of you having big A, little a is 1/2. So next, big B, little b with big B, little b, you would have a 1/4 chance to get little b, little b. Big C, little c with big C, you would have a 1/2 probability of getting big C, little c. Big D, probability is equal to 1/4 big D from a big D, little d cross. And then you would have a 1/2 chance to get big F genotype from a big F, little f cross. So this should all be equal to, let me calculate it real quick. One over 128, and that'll be your answer. So pretty simple right there. Not too much work with that. And don't waste your time trying to figure out the entire ratio when you can just figure out a specific combination. But in this situation, we have something different where the question is telling you to figure out the phenotypic ratio. So now we have to use the matrix method or the branching method, and I'll show you both of them as well. So with the matrix method, what we're going to do is we're going to start off with both of the genes independently. So big A crossed with a big A, little a. It'll be, for the phenotype, it'll be 100% big A whatever. And then the question is telling you for big B, little b with big B, little b, the phenotype proportion will be 3/4 of them would be big B whatever, and then 1/4 will be little b. So you would end up with a 3-to-1 ratio, which is essentially big A, big B, which is this one. And then you have one that is big A whatever with little b. And that'll be the 3-to-1 ratio. Now, unfortunately, the branching method for this problem, we're going to do another problem, obviously, but with this one is quite simple. It's exactly the same thing because we only have one node in the A gene. Well, we only have one product from the A gene, so which is just big A whatever. And because we have two products in the B gene, this node is going to get two different branches. And then you'll get an answer of 3-to-1. But let's do another one where the question is asking you for genotypic ratios. So if we're trying to find the genotypic ratios, crossing big A with big A little a, we would have a half chance to get big A, and a half chance to get big A little a. And then for the B gene, we would have a 1/4 chance to get big B, 2/4 chance to get big B, little b, and then 1/4 chance to get little b. And what I will tell students right now is that the reason why I didn't write this down as a 1/2, if you wrote it down as a 1/2, you would mess up your ratios, your final answer as your ratio. So what you want to do is you want to make sure that for each of these genes, like gene B for example, they all must have the same denominator. If the question was asking you the genotypic frequency and you're multiplying it by 1/2, that's fine. But if the question is asking you something like genotypic ratios, you must make sure that from the B gene and all the frequencies in the B gene are all have the same denominator, okay? So same thing with the A gene. All of my products from the A gene, they all have the same denominator, okay? So right now what we're going to do is we're going to multiply across. So big A, big B, we would have a 1/8 probability, big A, big B. Then we're going to do big A with big B, little b. 2/8 big A, big B, little b. And then we're going to do 1/8 big A, little b. Hopefully that makes sense. And then big A, little a with big B. We're going to have a 1/8 big A, little a, big B. We would have a 2/8 big A, little a, big B, little b. And then we would have a 1/8 big A, little b. So our final ratio from this is you make sure that all your denominators are the same at the end over here, and then you just write down your numerator. So 1 to 2 to 1 to 1 to 2 to 1. So that'll be your genotypic genotypic ratio. So let's do the branching method now. From the A gene, we have a total of 2 products, so we're going to write it down as 2 nodes, big A, half chance, and half chance to get big A, little a. And then because we have 3 products from the B gene, we're going to write down 3 branches for each node on from the A. So what we're going to do is each of these nodes gets 3 branches. One of them is going to be 1/4 big B, 2/4 big B, little b, and then 1/4 little b. Same thing over here. 1/4 big B, 2/4 big B, little b, and then 1/4 little b. So we're going to multiply them across again. So 1/2 times 1/4, you would have a 1/8 chance to get big A, big B. 2/8 to get big A, big B, little b, and then 1/8 big A, little b. Same thing over here. 1/8 chance to get big A, little a, big B. 2/8 chance to get big A, little a, big B, little b. And then 1/8 chance to get big A, little b. So hopefully that helps you out for all these crosses. And we're gonna go in tomorrow or some other day, whenever you watch it, to go over the probability rules of why we're actually multiplying these across. But yep, that's it.